So we've been able to identify patients that are more applicable to these therapies, but we've also been able to identify patients that would not respond to these therapies in the first place, and so these patients would therefore no longer have to go through these hard chemotherapeutic treatments, whereby we now know that they would no longer work in the first place. My name is Pieter Eichhorn. I'm a group leader at the Curtin Medical Research Institute and a professor at Curtin University. I had a very unique lead up to where I am today. So basically, I did my undergraduate degree at the University of Western Ontario, and then I decided to do a PhD. And I ended up doing a PhD at Newcastle University upon Tyne in Northern England, where I ended up working with a leading developmental biologist called Tom Strachan. Following my PhD, I decided to pursue my interests further at the Netherlands Cancer Institute in Amsterdam. And I got extremely lucky when I went there. I ended up working with a leading professor called René Bernards, who was actually doing some of the world's first transgenic studies where he was knocking down individual genes. At the time, nowhere else in the world was doing this, and we were at the front line of being able to knock down individual genes and then looking at mechanisms of resistance to targeted therapies. And through this basic biology, I then wanted to pursue more clinical interests, and I ended up going to work with Jose Baselga at Vall d'Hebron in Barcelona, where he was the #1 clinical oncologist looking at breast cancer research. And through all the techniques that I had learned in René's lab in Amsterdam and analyzing the patient samples that Jose had access to, we were able to do some transformative work looking at mechanisms of resistance to drugs that were currently being used in breast cancer. Following this, I ended up at Harvard for a few years, again pursuing my interests in looking at this translational research. And then I decided to pursue different interests at the Cancer Science Institute of Singapore, where we're looking more at how RNA itself was influencing these mechanisms of resistance. And for the last 8 years, I've been in Curtin University, working at the Curtin Medical Research Institute. The focus of my research for the last 20 years has been looking at mechanisms of resistance to targeted therapies, primarily focusing on feedback loops, rather than looking at the individual mutations inside the tumors and how these influence therapeutic responses. These feedback loops are primarily dependent upon transcriptional changes, where they might influence how individual pathways inside the cell react to these inhibitors, rather than looking at how the individual mutational background of these tumors might influence the inhibitors themselves. Through this work, we've identified a number of these feedback loops that influence how these inhibitors work, and we've been able to translate that back into the clinic and show how these affect patient responses inside the clinic. So we've been able to identify patients that are more applicable to these therapies, but we've also been able to identify patients that would not respond to these therapies in the first place, and so these patients would therefore no longer have to go through these hard chemotherapeutic treatments, whereby we now know that they would no longer work in the first place. Most of my work is primarily funded through the World Cancer Research Fund, through the Australian Centre for RNA Therapeutics and Cancer, and through the Cancer Council of Western Australia. My collaborations currently are highly involved with the Walter and Eliza Hall Institute, where we're identifying novel inhibitors targeting deubiquitinating enzymes and how they affect breast cancer and lung cancer progression. We're also working very, very closely with the Australian Centre for RNA Therapy. We're identifying novel ways to target RNA in cancer, and then we can utilize these techniques to inhibit the RNA in cancer and looking at ways to increase the therapeutic responses in these patients. Of course, the majority of work that we do looks very, very closely at overall patient responses and how patients respond to targeted therapies. But in reality, all of our work that we do is looking inside the cells itself, looking at how the 20,000 or so different genes, the variations of thousands and thousands of proteins, and how they all interact with one another and how that cancer cell evolves over time. So when we look at the results, trying to identify hypotheses of what's happening inside those cells, and then looking to see that those hypotheses end up true, and getting that result for the first time is an amazing experience to say, wow, this is what's happening. This is what's really happening inside the cell, and this is what's causing these cancer cells to keep on proliferating. To get that result, to be able to look at that result and realizing that no one else in the world has seen results like that you're the first one to know that, is an unbelievably amazing feeling. Furthermore, the passion of what that comes through is looking at how this eventually affects patients in the clinic and realizing that you are really making a difference towards these patients. My interest outside of work, I always find this a very unique and interesting question. I always find that it changes as you age. When I was younger, I used to play American football. And of course, as you get older, the knees start to go and the elbows start to go and the back start to go. So you no longer can be able to do those physical activities. The majority of my interests now are actually anything to do outside. As long as I'm outside of the house doing stuff, and that can include gardening, to going for long walks in the hills, to going for swims at the beach, any of those activities I'm really highly interested in. And one of my great passions is to take the dog on a Saturday morning and to go for these 20-kilometer walks in the hills, and just being outside in nature is one thing that I really, really love to do.